Okay, we're going to talk about the structure of DNA, and we're also going to talk about replication. So, a few key concepts that we need to review. Replication of the DNA occurs during which stage of the cell cycle? And that is S phase. Keep in mind, another word for replication is synthesis. What are the four nitrogen-based pairs that make up DNA? We have adenine, always pairs with thymine, and we have guanine, always pairs with cytosine. What is the sugar in DNA called? It's where the D comes from in DNA. And it is deoxyribose. So that D right there. How many strands are there in DNA? DNA is a double strand, so there are two. And what is any change in the DNA called? We haven't touched upon that yet, but you should remember this. This term comes up quite a bit. This is called a mutation. We'll talk more about this in transcription and translation, but know that any change in our DNA is called a mutation. Some mutations are good, some are bad, some have no effect. Okay, vocabulary we need to know. Replication is to make a copy. And again, synthesis is almost the same word. Synthesis means to build. So when we talk about DNA replication is being synthesized, we're building a new DNA. Okay, so a nucleotide is the building block of DNA and RNA. So that is a building block. Of DNA and RNA. Double helix is the twisted ladder shape of the DNA. Okay, an enzyme is a protein that helps reactions occurs. And we've already said that a mutation is any change in the DNA, which is why we spell check during the G2, is to try and avoid mutations. Okay, so here's a structure of the DNA, and a nucleotide is one of these sections. Oops, that's a nucleotide. We have phosphate. This is the phosphate end. Let's do this one. That is the sugar. And again, we're talking DNA, so it is deoxyribose. And This part right here is our nucleic base or our nucleic acid. Two together are our base pairs. And you'll see that the deoxyribose of 1, or the phosphate of 1, binds with the deoxyribose of the one above it. The deoxyribose, the sugar, and the phosphate make up the backbones. And the sugar make up our backbone. The base pairs make up the rungs of our ladder. So if you look, you can see the ladder and you can see the rungs. The base pairs make up the rungs. And this is a hydrogen bond. Hydrogen bonds are strong chemical bonds. Between the two base pairs. Okay, so to look at the replication, in step 1, this is step 1, the DNA straightens out. It lengthens and straightens. In step 2, the enzymes break the hydrogen bonds. Between the base pairs. Okay, so as it breaks the ladder, then free nucleotides bond to the open ends. This is step 3. So here the brown G is going to match with C, so a green will come in here and bond right here. The green will have a brown come in and bond right here. The red will have a blue come bond right here. And same for the other side. And this will keep going. Bonds to the open DNA. This process is going to continue until there are 2 exact copies of the DNA. And then the DNA is going to twist back up. But you can see as the free nucleotides came in and bonded with the left side and bonded with the right side, because the A always matches with T and the C always matches with G, then we wind up with 2 complete sets of DNA. So at the end of the S phase, at the end of interphase, going into mitosis, we have 2 complete sets of DNA. At the end of mitosis and at the end of cytokinesis, we have 2 exact copies of ourselves.